Hi everyone, this is Ben Dichter, and this is the NWB tutorial for reading NWB data in MATLAB. So we're going to be walking you through how to find a publicly available NWB dataset, in this case single neuron spiking data for a visual task, how to understand the structure of the NWB file, and then conduct some simple analyses. So for this demo, we're going to be using a data set published by Chantravarya very recently out of the Ruchasauser lab, and they recorded electrophysiology activity in human subjects from the mediotemporal lobe while the subjects performed a visual recognition memory task. So this data is actually publicly available on the DANDI Neurophysiology Data Archive, and this is where we'd recommend that you share NWB files with each other. So I'm going to show you how to download this data. So first I go to dandiarchive.org, and then I go to public datasets. This is dataset ID 4, and there's a particular file that I like. So these are all of the subjects. You go into a particular subject, you see the sessions associated with that subject. In this case, there's just one session associated with the subject, and you click the download button and you should be able to download the data. I've already downloaded, so I'm not going to continue and download it again, but that should work for all of you. It's publicly available. And if that was too fast for you, here are some diagrams for how to do that. So the first thing we're going to do is install MatNWB and make sure that it's set up with the right schema to read this NWB file. So this file was created with a bit of an older schema. So we're going to have to do a little bit of extra work to set up MatNWB in order to read it. So first we're going to clone the MatNWB code base, and then we're also going to clone the NWB schema repository. And once that's cloned, we need to go into NWB schema and check out this version 2.1.0, which is the version that was used to write these NWB files. Once we've done that, we can go into MatNWB and copy in the schema that we've now checked out into the MatNWB directory, and then add everything to the path, and then finally run this generate core function, which will automatically generate the API based on this new updated NWB schema. So this is a bit, it's not as user-friendly as we'd like it, and we're working on getting this to work so that it can kind of automatically update the schema based on files. For newer NWB files, these types of steps wouldn't necessarily be necessary, but for this one, you have to do a few extra steps just to set up your environment. So once you've done that, you'll want to read the NWB file. And so to do that, all you need to do is run Nwb Read. Here I've directed the read to where the file is saved on my own computer. You'll have to change that on your own. And I'm outputting it into this NWB object. So I'm going to just run this cell, and so that's chugging along. It's kind of messing up just because those files already exist for me, but let's see here. Okay, so once you run this, you can see a lot of the attributes and metadata already within this printout of the NWB object. So you can see that there are two elements in acquisition, and you can see that it's from the Ruchasauser lab. You can see that there is stimulus presentation. There's an object here. So another way that you can that you can explore the NWB file is by running util.nwb tree. This will pull up an interactive hierarchical view of the file, so you can press these arrows and expand down and see the hierarchical structure of the file, and get a sense for what you can explore using the API. So let's first look at stimulus information. So first we'll just do nwb.stimulus presentation, and this holds all of the stimulus objects. In this case, it's a set with just one element in it. It's called stimulus presentation, and it is an optical series object. So optical series is the data type for expressing visual stimuli that are presented to a subject. So we use the get method on stimulus presentation to get this optical series object that's called stimulus presentation. And now the printout of this, since I have left out the semicolon again, is all of the attributes that are in this optical series object. So there's a distance between the eyes of the subject and the screen. There's a field of view, and there's the data, which represents the actual images in RGB or in grayscale that are shown to the subject, and timestamps, which indicate the precise times that these images are shown. So let's take a look at the images that were shown. So we can just do nwb.stimulus presentation, get the stimulus presentation object, and then .data. And here it doesn't actually return the matrix. What it does instead is return something called a data stub, which is sort of like a window into the data that's stored on disk that allows you to read this data, we call it lazily. What that means is that you might not need to read every data element in this data set. And so this allows you to read only specifically the data elements that you need for your specific task. And you do that with the load method, which I show down here. So here we're going to take image index 31. And our data set, we can see what the size of it is here. This is 3 for RGB, and then 300 by 400 is the size of the image, and then there are 200 images. So this syntax here, this way of calling load, says I want RGB, the first dimension I want 1 through 3, in the second dimension I want 1 through 300, the third dimension 1 through 400, and then in the last dimension we just want this image index. And so this will pull out precisely the data that we need, and we don't need to read the rest of this data into memory. So then we can load this in, and then there are a few things we need to do just to visualize it with IMShow. It's just read in the wrong orientation, and then kind of an oddity of this data set is that the images are stored in BGR, not RGB, so we need to read this last color dimension in reverse order. So we just permute it, flip this orientation, and then finally we can visualize the data set, this image. Okay, so here is the image. Let's see if we just change this to 32. We should be able to see that image as well. Okay, there you go. Now, if you do want to read the entire data set, all you need to do is call this load method without any input arguments. So if you just say load, it will automatically read the entire data set. And that's what we want to do here for reading in the timestamps. So now that we have looked at the stimulus information, let's take a look at how the neural response relates to the stimulus. So we're going to do kind of a quick PSTH and raster based on the data in this NWB file. So for this, we're going to need to select a specific unit. I'm using these little sliders here so we can easily change it if we want to. The number of seconds before the time align point, the number of seconds after the time align point. So these are our parameters. Now, spike times are stored in nwb.units.spiketimes and spiketimes index. So this is an index column that we're going to get into more detail about this later. But essentially, this allows us to efficiently store ragged arrays. So these are like matrices where the rows of the matrix have a different number of elements in every row. So this is something that you would use a cell array to handle this type of information, or a struct to handle this type of information in memory in MATLAB. We're reading it from the NWB file, and this actually with read index column will read a specific row of this indexed column that is spike times. So it will give us all of the spike times for a single unit. Then we are going to loop over all of the times that we have now and align our unit times to that to the stimulus presentation time, and then trim them to be right around that stimulation presentation time, and then add it to our results. So our results are going to be a cell array where each element of this array has the trial aligned spikes for that trial. And then finally, we're going to do a quick and dirty peristimulus time histogram with this data. So let's go ahead and do that, run all of this. Okay, and so here you go. So time 0 is the stimulus time, and you can see that this unit increases its firing rate a bit around just after the stimulus time. And if we go up here and we change the unit, let's go to unit 10. It's going to recompute all of this, and if we scroll down, okay, we see that this one also increases, but not so much. If we go change this again to unit 8, you can see that this one decreases its activity just after the stimulus onset. So there's a lot more you can do with this data set. I mean, I'm really not doing any analysis that has to do with the actual content of the images that were shown or the memory task. This is really just enough to get you started and show you how to read data from basically any NWB file. So I want to point you to further information about this data set. So this data set was published along with an extensive open analysis that is written in both MATLAB and Python, and it's publicly available on GitHub. So if you're interested in this data set, I really encourage you to check out this repository and see the analysis that they put together. I think this is a really rich data set, and there's so much more that could be done with it. So, so this is a really good, I think, launching point for someone who's really interested in secondary analysis of existing data in NWB. But this is just one data set of many that are available already on the DANDI Archive. So if you go to the DANDI Archive, you can go to public data sets, and lots of really interesting stuff here. You can go ahead and use the search bar. There are several data sets already that have been published from NeuroPixel recordings. So that's really exciting, and you can check out those. They're high quality data sets as well. The only reason I'm not showing you a demo with those is that the data tends to be pretty large. So I'm showing you one that's a little bit more compact and a little bit easier for everyone to handle just in a demo setting. But yeah, go ahead and check out those. And if you create NWB files that, that you think you would like to make public, go ahead and use the DANDI Archive to do that too. Make sure you check out the DANDI breakout session later to learn about how to do that. Okay, so that's kind of the workflow for how to take public data and start to understand what's in it and analyze it. Please let me know if this works for you, if you have any questions. Thanks.